Hi, it's Mr. Bennett, and in this video we need to talk about enzymes. Enzymes are proteins, and what they do is they speed up chemical reactions so you can stay alive. Now, the technical term you see up here, catalytic proteins that speed up chemical reactions by lowering the energy of activation. Calm down, I'll teach you what that's going to be. An enzyme is like this rocket on this little turtle right here. This turtle could move forward slowly by itself, but if you light this rocket, it's going to move a whole heck of a lot faster. That's what an enzyme is like. An enzyme allows your body to do chemical reactions much faster so you can stay alive. So let's hammer out some vocab terms first. First off, a catalyst. A catalyst is something that's used to speed up a chemical reaction. And how it does it is by lowering the energy of activation. I'll teach you what that is in a second. So a catalyst is anything that can speed up a chemical reaction. You actually have some of these in your car. Your car has what's called a catalytic converter that does a chemical reaction that gets rid of the smog in your car exhaust. Now, your catalytic converter is not an example of an enzyme. An enzyme is a catalyst that's made out of protein, and it's found inside of your body. So what's this energy of activation thing that we're talking about? Chemical reactions require some energy to get them going. So any chemical reaction you can think of, it requires a little bit of energy to get that thing going. The amount of energy that it takes to get that chemical reaction going is called the energy of activation. Well, what an enzyme does is it lowers the energy of activation so you can get that chemical reaction going with much less energy. So here what you're looking at in the top. Here is a picture that shows this is the chemical reaction. And this wall here that we have to get over, that's how much energy we have to add to the chemical reaction before it starts going. Well, this line right here shows you how much energy you need to get it going once you add an enzyme. And so what you're looking at is enzymes make it so you need a whole lot less energy to make a chemical reaction work. The more common way you'll see this picture is right here. The red line represents how much energy you have to make a chemical reaction happen all by itself. The blue line shows you how much less energy you have to make that same chemical reaction happen with an enzyme. So I just threw a bunch of big words at you. Let me give you a simple example of an enzyme at work. This is sucrose. This is table sugar. This is the stuff that you put on your cereal in the morning. Now, sucrose is made out of carbon, hydrogen, and oxygen. Now, what happens is sucrose can be broken apart into carbon dioxide and water. You can take some sugar and you can break it apart and make carbon dioxide gas and water. Now, this naturally happens. If you have a bag of sugar and you leave it out, it'll slowly break apart, making carbon dioxide gas and water. But this takes years. It takes years because you have to add energy to the sugar in order to make it break apart. Well, your body does the exact same thing. Your body can break sugar apart into carbon dioxide and water. It's called cellular respiration. Your body does it in about 5 seconds. Now, it does this by using enzymes. And so what the enzymes do is it makes it so you need a lot less energy to make this same chemical reaction happen. This reaction of sugar being converted to carbon dioxide and water, this will happen all by itself, but your body needs it to happen a lot quicker, so you use enzymes to make that happen. All right, so before we can talk more about enzymes, we got to hit up some basic enzyme vocabulary. This is basic vocab that's used whenever someone's talking about an enzyme. There are two main types of enzymes out there. You have anabolic enzymes and you have catabolic enzymes. Let me explain what they do. An anabolic enzyme is an enzyme that'll take two things and put them together. Now, in your last video, we talked about macromolecules, and macromolecules are made out of monomers. Well, all the macromolecules in your body are made by an enzyme. But there's an enzyme that will take two of these monomers and it will put them together to make the macromolecule. So that is an anabolic enzyme. How I remember anabolic is the proper name for steroids or anabolic enzymes. Anabolic means to build. Someone who takes steroids, they're going to build some muscle. They're going to shrink something else, but they're going to build some muscle. And so that's how you can remember anabolic. Anabolic builds. The other type of enzymes we have is we have catabolic enzymes. And what catabolic enzymes do is they tear stuff apart. I think cat, tearing things apart. Catabolic cat. So enzymes make things and they break things. The enzymes that make stuff are the anabolic enzymes. The enzymes that break stuff are the catabolic enzymes. Here you can see in this animation this picture, that we start out with one molecule. The enzyme grabs it and breaks it apart into two molecules. So all enzymes fall into these two categories. Every enzyme is either an anabolic enzyme, it makes something, or it's a catabolic enzyme, it tears something apart. Now, a couple other terms we got to talk about is we got to talk about substrate and we got to talk about products. Substrate, good term for this is ingredient. The substrate is the ingredient that the enzyme is using. So this enzyme here is using these two molecules A and B. These are the substrates. And then it takes these two substrates, puts them together, and makes a molecule AB. This would be the product. So what the enzyme uses is the substrate. What the enzyme is making is the product. Over here with this enzyme, with this catabolic enzyme, it starts out with the substrate sucrose, and then this enzyme breaks it apart and makes glucose and fructose. So this enzyme has the substrate sucrose. This is what it will bind to, and this enzyme spends all day making glucose and fructose by breaking apart sucrose. So how do enzymes work? How do enzymes do all this magical stuff? Well, the first thing an enzyme needs to do is it has to physically bind to its substrate. It needs to physically bind to its substrate. And the place on the enzyme where the substrate binds is called the active site. So what you're looking at here, these red stars, these represent the active sites of this enzyme. Here's the substrates. They come in and they actually physically bind to the enzyme. When they physically bind to the enzyme, what the enzyme will then do is it either holds the substrates really tight together, forcing them to form covalent bonds, and so therefore they're joined together. That would be an anabolic enzyme. Or what the enzyme will do, if it's a catabolic enzyme that breaks something apart, the substrate binds to the enzyme, and then the enzyme then actually physically breaks it apart. So all enzymes, in order to work, they have to bind to their substrate. They have to bind to their substrate. And if the enzyme can't bind to its substrate, it doesn't work anymore. So the enzyme binds to its substrate, and then it either joins those substrates together if it's an anabolic enzyme, or it tears them apart if it's a catabolic enzyme. Last thing we need to talk about is we need to talk about some characteristics of enzymes. So these are some things that all enzymes have in common. First off, when we name an enzyme, and your body literally has 75,000 different enzymes in it. Think about that for a second. 75,000 different of these proteins that do the chemical reactions that keep you alive. When we find a new enzyme, we usually name it after the substrate that it works on. So you have an enzyme in your body that will break apart sucrose, table sugar, so you can digest it. Well, the enzyme that we use to break that apart is called sucrase. Here's a hint. If you ever see something in biology and it ends in ASE, you're probably talking about an enzyme. So sucrase, lactase, whatever, galactase, these are all different enzymes. So if it ends in ASE, it's probably an enzyme. And the first part of it is telling you what it works on. So the enzyme sucrase breaks apart the sugar sucrose. The enzyme lactase breaks apart the sugar lactose. Now, one thing about enzymes is they are substrate specific. Sucrase cannot break apart lactose because lactose will not bind to sucrase. Only sucrose will bind to it. So for each chemical reaction you want to make happen, you need to have an enzyme that'll do it because each enzyme will only bind to one type of substrate. Next thing we got going on. Enzymes work faster when it's warm and they work slower when it's cold. This graph right here, what you're looking at, temperatures from 0 to 37. 37 degrees Celsius is body temperature. This is showing the average enzyme in your body. The reason why your body temperature is around 37 degrees is that's the temperature where your enzymes work the best. If it's colder than 37 degrees, your enzymes slow down and they don't work as fast. If it's warmer than 37 degrees, they actually don't work as fast either. So for every enzyme, there's a sweet spot. There's a temperature sweet spot where that enzyme will work better. As it gets warmer and warmer, that enzyme will work faster and faster until eventually it gets too hot and then it actually kills the enzyme. I'll talk about that in a second. But for the most part, enzymes work faster at warm temperatures and much slower at cold temperatures. This is the reason why you put your food in the refrigerator. Your food has bacteria on it, and these bacteria are going to use enzymes to break apart your food and make it go bad. Well, by putting your food in the refrigerator, you slow down the enzymes of the bacteria that make your food go bad, and that way your food doesn't go as bad quite as quickly. Other characteristic of enzymes, enzymes are not destroyed during the chemical reaction. They're used over and over and over again. They are recycled, and they just spend all day, every day doing their chemical reaction. And then the last thing to talk about, as mentioned, enzymes are destroyed by high temperatures. An enzyme will work faster and faster until you hit a certain tipping point, and then it gets too hot and it actually breaks the enzyme apart. This is the reason why if your body temperature gets above 106 degrees, you're probably going to die. Because at 106 degrees, your enzymes stop working because they're actually being destroyed by the high heat. The other thing that can destroy an enzyme is big changes in pH. Here you see 4 different enzymes here, and you see pH. So this enzyme, chymotrypsin, it works best at a pH of 8. If you get more basic than 8 or more acidic than 8, the enzyme stops working as well because it gets destroyed by if it's too acid or too base. Here you see another enzyme, pepsin. Pepsin works best at a pH of 2. If the pH gets higher than 2, it stops working. So every enzyme has a pH sweet spot as well, and if you change the pH too much from that sweet spot, it stops working. So that's enzymes for you. They make stuff, they break stuff, they speed up chemical reactions, and without them, you'd be dead.